So I can't hear you.
Yes, sir. Sir,
Hello.
yes, now we can hear.
Uh, uh,
We
so
didn't hear you from the beginning.
okay. So then I'll repeat it entire aspect because I have almost used more than 10 to 12 minutes. Fine. So dear students, if you remember, last time we came across with a practical that is minimum inhibitory concentration of a sucrose solution for yeast activity, and at that time you simply prepared a write up and submitted it as a part of your submission. But actually, how that particular practical maybe get performed, that part of discussion was remaining. So what I can do now, I'll just give you a brief idea about in what way minimum inhibitory concentration experiment may be get performed. And for that purpose, what you need two different types of a culture media. Basically culture medium in which microorganism like as a bacterium or fungus it is grown, that is considered that is selected for testing the minimum inhibitory the concentration. Now our experiment is related with you can call as a testing the sucrose concentration. After one specific concentration level, the yeast activity it may be get inactivated. So in case of a beverage industries or it may be in case of alcohol industries, role of yeast is very important, and its nutrient use that input in terms of a concentration should be standard one. And from that point of view, such type of certain test they may be get performed, and at what concentration this yeast is showing best result, that may be get checked. So dear students, the practical title is the minimum inhibitory concentration of sucrose as a chemical compound against the microorganism, more specifically yeast. And for the purpose, two culture media were used. One culture medium, basically for fungus there is need of a culture medium that is called a subarrowed medium, but subarrowed medium without sucrose, that gives rise to diluent solution. You can say that important ingredient of a subarrowed medium is of course sucrose when we culture the microorganism like as a yeast. But here what concentration is suitable, that has to be get tested. Below to that particular concentration, that is minimum concentration, if it is there, and microorganism cannot be grow. But in such cases what happens, the level at which microorganisms activity it is started to get affect, that is called as a minimum inhibitory concentration. So in most of the industries, this type of concentration, if that compound which is essential for the growth of microorganism, if it is in excess quantity and in enough concentration, at that time microbes they starts to grow their and they damages the product. But in industries what precaution is taken, the concentration beyond to which microorganism cannot survive, okay, such concentration it is get adjusted, and in this way the product it becomes much more quality product. So in this way the industries like as jam, jelly, syrup, in which you might have seen that products they are highly concentrated, and if to just make it a technical adjustment, so if someone technical person reduces the concentration of a sugar there, it may be definitely invite the activity of a microorganism. So in case of a quality assurance cell of these industries, they are very very cautious about testing the product, and therefore one of the interesting mechanism what we are going to study today, that is minimum inhibitory concentration of a sucrose. And for the purpose, experiment was set up with the reference to three basic test tubes, in which one test tube is consisting the stock solution. Stock solution means it consists almost 80% of a sucrose over there. So, but in this particular stock solution we are not putting any diluent, that is a concentrated nutrient broth we have selected, which consists of sucrose. And of course this is only 10 ml we have selected, and it in this solution we are not putting a yeast for activity. Second one, we have selected the 10 test tube in such a way that it consists only diluent, it is not consisting any sucrose, and in this particular test tube we are not putting a, you say that a yeast also. So in both the test tubes when yeast it is not given, okay, at that time the test tubes they are used for you say that a checking your results actually, but experiment it begins with a blank. You know that calorimeter reading it may be get possible only once you set put your test tubes over there in a blank. So the test tube in which there is only diluent and no yeast, neither concentrated solution, that is referred as a blank test tube, okay. So to adjust 0 or to create a 100% transmittance, it may be get used. Whereas the another test tube in which there is a only, you say that a presence of a concentrated one. So this concentrated solution it will be treated there as to see the positive effect of positive activity, or is simply called as a positive control. This may be gives, as it is absolutely higher concentration, it help us to know about the how in various concentration fungus is responding, so that it is referred as a positive control. Whereas in case of negative control, there is no as such sucrose concentration that is in a broth, instead of it is only a diluent, just like as a, you can call here as a nutrient diluent over there, or diluent solution or subarrowed agar medium. So in this way in blank there was no yeast now, but here we are putting the yeast in a concentration of a 0.1 ml. of its population. So yeast is added in a diluent, and this is referred here as you can call positive control. I'm sorry, very first one it was called as a negative control, and now where yeast it is added, it is called as a positive control. So positive control, negative control, blank, these are the three basic test tubes what we have to first of all make. And of course after that the experimental test tubes are test tube number 1 to 8, in which the concentration it may be varied at 10%, 20%, 30 to 80%. In this way, if it is an antimicrobial compound, or if it is an antifungal compound, or if it is any other, you say that antibiotic, at that time concentrations may be it may be in molar solution, it may be in ppm solution, or it may be in varied way micro quantity solution may be there, if you want to test the antibacterial compound or antibiotic in that way. So at present we are selected in percentage solution. Now how percentage solution is prepared? If for every broth it is not made in a 10% or 20% in that way, we have to dilute that one, and accordingly it becomes 10 or 20%. Say for example, if you put 1.25 ml of a concentrated broth, or simply called as a suborowed solution, or suborowed nutrient suborowed culture medium, which consists of sucrose. So in that, if you put a diluent of 8.45 ml, it gives rise to, I'm sorry, it may be 8.75 or 8.45, it gives rise to total 10 ml of a volume, final volume 10 ml is there. So in this way, once the test tube is prepared, its volume should be 10 ml. So combinations are made in such a way that when we add a concentrated solution and diluent together, final volume should be 10 ml. Accordingly it gives rise to values like as 10%, 20%, 30 or up to 70%. And each test tube it will be afterward added with 0.1 ml of a solution of you can say that yeast culture. Ultimately this particular solution we have to keep for the incubation. So more often what is done, the incubation time duration which is given, that is more than 24 hours. So after 24 hours these test tubes they are taken out, and the readings they are calibrated on, you say that a optical spectrophotometer, or it may be on the colorimeter. So when the optical density or OD it is taken, of course for that purpose a positive a blank, you have to you start with a blank, then you start with a positive control, you start with a negative control, and negative control more often it is recommended that should be carried out at the end actually, because it is highly concentrated one. So once you start with a blank, next one is a negative control, you have to actually go, because it doesn't consist any sucrose over there. So readings they are getting in a proper order also in fact. So in this way, the once reading are obtained, from these readings we can conclude that at what particular density, you can see here the density which is very optical density, of course it starts to get reduced at a concentration of almost 50 or 60% if I'm not wrong. So 60% sucrose solution, that is the point where the concentration it is getting affected, that fungal activity is getting affected actually over there, it doesn't show a much more significant growth over there. So this is referred actually as a minimum inhibitory concentration. So the point at which it begins, after that even at 70% it remains as it is over there. Okay, so that is 70%. But later on again what happens, that fungus if concentration it is exceed or almost 80%, it has little bit short activity. But you have to remember that one, if the point at which fungal activity is affected, if that particular concentration is selected for manufacturation unit, definitely it prevent the further action of a microorganism, and our product it will prevented from the any spoilage. Therefore from such experiments of minimum inhibitory concentration determination, we can conclude that this type of concentrations are really very important to prevent our product, or to make our product of a good quality, and it should be free from the microbial activity. So dear students, this was the part of a practical what we have performed in previous week actually, and due to certain technical reason it was not possible to discuss with you, but even then you did very nice job, and you have submitted your that assignment, or you simply called as that practical write-up over there. So after this, actually today what I have planned to discuss with you is another experiment that is called as perhaps this is the last practical of microbiology section, and after that we are going to, in fact overall practical, it is a last one. And of course we are going to discuss later on about submission of your entire journal now. Yeah, so far you have submitted daily basis certain fractions of your write up, but now since from the 7th of December onward your practical exam is going to commence, that perhaps that notification it will be issued to you shortly. So your journal should be ready now, and for that purpose you have to first of all attend the today's content, and after that you try to scan entire journal together and give it to us according to the schedule. How to submit, I'll inform you shortly also. So today's practical is about study of antimicrobial compound, and here you know that there are various chemical substances existing in nature or natural product. It may be in plant leaf, or it may be in rhizome, or it may be in any other organ, and from that extract generally what we do, we try to prevent the activity of microorganism which is hazardous or which may be harmful or pathogenic in nature for the plant as well as for the animals. So what we do, based on the very first practical, you know that microorganism can be get isolated. Okay, now next part is that this microorganisms activity may be get tested in such a way that the isolated microorganism has to be get brought in contact with a inhibitory compound. Now how it is done? It is done by means of a compound of our interest, which we suspect the compound what we feel is consisting a antimicrobial actions showing compound or antagonistic compound over there. So such compound solutions, they are prepared or extracted, and after that what is done, a petri plate it is selected, and on this petri plate what you have to do, you have to, by using the in theory you perhaps came across with a concept called as inoculation of a microorganism or isolation, pure culture, you might have seen that one. So in pure culture you know that there is a strict plate method. So microorganism of a interest, it is present in the form of a broth or in the form of a colony, right? So in this way, by means of a strict method, you can just apply on the petri plate which consists of microorganisms culture medium. So on this you just apply the strict way, so microorganism this starts to show their presence after few hours or few days of inoculation. So here microorganism this starts to grow actually. Okay, so this is the way how we can isolate by them by means of a strict plate method. But there is another method in which culture it is poured and it is get sprayed on this entire surface, that is called as a petri plate pouring method. So strict method and pouring method, these are the two ways of so that a isolation of the microorganism. Now same thing what we are doing here in today's practical, on a petri plate we are applying the microorganisms growth first of all, but this growth it is not allowed overall growth, but you have to pour it, inoculation has to be done. So yeah, at present you just forget about these two points, they are the control. What you have to do, you just apply the strict plate method like this one, zigzag microorganism has to be get inoculated. That is the one way. And second one, on petri plate you can pour the microbial culture and spread it on this uniform surface. So either by means of a strict plate method or by means of a pouring method, microorganism is brought in contact with a leaf on petri plate surface. After that what we have to do, you have to select a small discs are there. So these are, can you see here, these are the discs over there. So antimicrobial compound like as say for example antibiotic streptomycin is there as a control. Say for example streptomycin, if you want to check the activity for that particular fungus or for that particular bacterium, what we are doing, that streptomycin's solution has to be get prepared, and in that this disc, paper discs have to get dipped. So the known concentration of a antimicrobial activity in that solution, we are dipping the disc, and this disc it is further put on the previously inoculated petri plate. Similarly, the disc which is present there, of course sterilized, it is made up of a sterilized Whatman filter paper. It is get punched into the circular disc, and such discs they are used for testing the microorganisms, antibacterial compound or antifungal compound. So in case of a control, positive control, what we can do, the disc is simply dipped into the solution in which streptomycin it is prepared. but not streptomycin. It means it is aqueous solution. Suppose if it is there, or saline solution if it is there, in that solution only the disc it is dipped and it is brought in contact with the surface. So here there is no antimicrobial compound, but here is an antimicrobial compound. Now just have a look for the another compound. If you are interested in a plant material, in that case what is present, turmeric say for example, or it may be the ginger, or it may be the clove, whatever may be it, from that a known concentration of a solution it is prepared, and in that solution disc is dipped and it is brought in contact with a microorganism which is inoculated on this plate. Of course control it is without any kind of a antibacterial or antimicrobial compound. So in this way these two discs are placed. Now what happens after a few days of a inoculation? Just remember, this is the same petri plate, okay, this is the same petri plate in which, now this is the area at which now that student has not drawn it properly, but you have to remember, in fact this disc it is not this disc is has not to be get placed at such a corner or edge, it should be here or here, okay. So in this way it should be placed in proper way. So that is a little bit wrong, but please remember now, here suppose this was the disc of antimicrobial compound. After few days of inoculation, what happens, the microorganism which was present on the surface, it starts to grow its activity. That means if it is a fungus, it starts to grow anywhere wherever it is present, but as this antimicrobial compound it is present, the microorganism cannot reaches up to this central part of a disc, or close to the disc, means some portion it is remaining blank as it is without growth of a microbe. So this particular zone it is produced over there, its distance it is very shorter, more often it is in millimeters, okay. So how broader is the distance away from this disc for that microbial growth, that indicates, if the distance is more, it indicates that antimicrobial activity of compound is very best one. If distance is shorter, comparatively activity is lesser. So that concentration, of course activity is there, but concentration we can decide. So you have to prepare such a concentration in which during your research actually you have to prepare various concentrations, and from that a concentration in which a best of best result, that is maximum distance it is get achieved, such type of results you have to find over there. So dear students, study of antimicrobial compound by means of a method what we have used here, that is applying the disc, is referred as the disc diffusion technique. Now why it is called as disc diffusion technique? Answer is very simple. Now see here, this was the disc, now I have tried to magnify it. So disc it is kept here, and surrounding to that is a petri plate, and on this petri plate microorganisms they are growing everywhere, but toward the periphery of this particular disc, at certain distance, up to certain distance microorganism only grow, and after that it cannot reach toward the disc because there is a antagonistic compound. So this particular line, okay, the zone what you have observed from the periphery of the disc and toward the edge or margin of a microbial growth here. So this particular distance only, this line, I am talking about this line only, is referred as the, and this particular distance has to be get measured, and it is achieved by means of marking the petri plate from the downward surface, and you can say that this marking is in few millimeter, and once it is get measured you will find out that suppose there are 4 to 5 points are there. So in first case it was 2 millimeter, second made 2 millimeter, another one 1.5. so in this way for same concentration if you obtain these 5 different readings, okay, 5 petri plates were there, and in 5 petri plates in 2 plates 2 mm, in third it was 1.5 mm. So average what was that distance of a zone of inhibition has to be get measured. So in present case ultimately final distance is a 2 millimeter. So what it indicate, the zone of inhibition by means of a 2 millimeter was the experimental reading over there, and in fact another conclusion it may be get made, if it is a bacterial colony after 24 to 48 hours, when we observe this disc we will find it out that the disc they are having varied activities. So say for example if very close to that disc microorganisms are growing, that is referred as a less effective antimicrobial compound. If activity is that means if this distance it is 0.5 mm say for example, that is a less effective. Okay, so intensity of activity of compound, it may be sometimes designated by means of a plus for the less effective, 2 times plus, that is indicating moderate antimicrobial activity. Say for example 1 mm, there is moderate activity, and if it is a 2 mm, in present case it was reported, it is called as highly effective antimicrobial compound. I hope that this type of results may be possible only when you perform this experiment in at your own level only in laboratory. So dear students, what I supposed to tell you was this particular practical is very important, and from the point of a application view in your personal career, this particular practical, it may be helpful. So this was the part of a today's discussion. What I had done with you is that today's practical, if I'm not wrong, I have posted in just a minute. I have posted it into the your Google Classroom. I'm sorry. Okay, you just check your Google Classroom. Yeah, today's practical, it is posted. You just click and open. It consists of today's practicals discussion. So in this way there is a form, or you say that a Google document which is with you. You just click and open and refer it for writing purpose. If you are not having this particular, if you fail to get this one earlier, even I have showed given you a handwritten journal PDF file, even you can refer that particular file today, and it may be help you to write the things. So dear students, the today's practical write-up is like this one. Content, you just go ahead with writing the part, and after that you will get an idea. The blank pages are there, and on these blank pages you can illustrate the experimental results actually. So please, when you are drawing the diagram, here is a correction what I am suggesting you. So make a vertical hypothetical vertical axis if it is there. Here will be one control, here is a streptomycin disc, or here may be a control one. So it should be in proper way. It should not be toward the periphery, it should not be close to this margin, it should be somewhere away from this distance. So in this way, this correction you have to make. And after that second diagram is like this one. So of course the water may be positions of the disc are kept in a first diagram, same should be disc positions here. And after that you have to make a such a diagram. Here actually linings are given, they are indicating the microbial growth over there. So instead of that you can just make that surface darker, and in this way the disc, it will be more distinct, and in fact zone of inhibition, it will be recommend more distinct. And this is the third diagram actually. So first, second and third, these three diagrams you are supposed to draw, and toward the end these are the results. Basically conclusion what you can make, and what are the results you have to write it over there. So practical is very simple and easy, but very important from your skill purpose, what you gain at the end of this particular academic session. So I hope that you understood today's all instructions, and keeping hope that you will perform, you will write all this in proper way, and will submit it in a due course of a time in fact. So dear students, along with this particular use that a practicals discussion part, I have given here a attendance link, and you know this attendance link is meant for your practical submission only. So you now it is, as it is in a Google Classroom, it is safe, no problem. No one need to ask it, sir, where is that link. So you can definitely use it over there. Now question is that as your internal assessments of one of the student was going on at 8 p.m., it was not possible for us to conduct the lecture. In fact, since for the last few weeks exams are going on, and every time certain people's lectures are get sacrificed, at least one or two lectures may get sacrificed. In fact, today I had lost one more, and perhaps yesterday Nitin sir also. So instead of going for the practical at afternoon, and in fact I was waiting for your recommendations for regarding the revision, but no one has interested in revisions. I'm sorry. So I plan to discuss with you about the practical. I'm sorry. So though practical discussion is made at present, it will be definitely need certain time, and a link it is given to you. The moment at which you finish your practical content, you can submit your practical write-up. So link is there with you. Now another part what I want to say here is uh, about the lecture. On record there should be at least your presence or your engagement was is expected by the office of principal. So what is done, the attendance form has been prepared for yeah, attendance form has been prepared, and you are supposed to fill that attendance form and submit it. So what I'm doing now, I'm just providing you a attendance form for the lecture. Actually, though I have made here a practical discussion, but here what I can do, I can provide you a attendance form for this particular uh, session. It will be treated there as a session of your theoretical discussion. So I'm putting here the attendance form. You fill that attendance form, and after that you are free. So while filling this form, if you face there is no question and answers at all, but just a minute.
सर ये फॉर्म भी फिल करना है जो आप भेज रहे हो और गूगल क्लासरूम पे जाके भी फॉर्म फिल करना है प्रैक्टिकल का.
Listen, एक बार, just a minute, check this one first of all. Form is self explanatory, you will get an idea. ओके, फॉर्म पहले चेक कर लीजिए. जैसे आप ओपन हो जाता है आपको आइडिया आ जाएगा कि इस फॉर्म के अंदर आपको थियोरटिकल, ये थियरी का सेशन मॉर्निंग में अपना थियरी रहता है, तो थियरी के एंगेजमेंट के लिए आपको ये फॉर्म फिलअप करके देना है. ओके, सो दिस इज़ a टाइम ऑफ 9 to 9:48 सेशन, एंड अकॉर्डिंगली दिस विल बी ट्रीटेड एज ए थियोरटिकल अटेंडेंस. ओके, थियरी का अटेंडेंस ऐसा इसे कंसिडर किया जाएगा. नाउ जब आप गूगल फॉर्म में जाओगे, गूगल क्लासरूम में जाते हो, वहाँ जाने के बाद आपको जो फॉर्म मिलेगा, दैट फॉर्म इज़ रिलेटेड विद द प्रैक्टिकल. जो आप एक्चुअली प्रैक्टिकल करने के बाद जो लिंक आप वहाँ पे पेस्ट करते हो, उसके लिए वो फॉर्म दे दिया है. आई होप दैट यू अंडरस्टूड इट नाउ. समझ में आया बेटा? हेलो, कैन यू आंसर?
Yes, sir.
हाँ, आई होप दैट यू अंडरस्टूड इट. सो फॉर्म मैंने आपको भेजा है, चेक कर लीजिए. फॉर्म ओपन हो रहा है. इट इज़ ए मैटर ऑफ हार्डली 2 to 3 minutes ओनली. नथिंग इज आस्क वदर एज ए क्वेश्चन. सभी को इंस्ट्रक्शंस प्रॉपरली समझ में आ चुके हैं क्या क्या करना है. सबसे पहले थियरी का कंसिडर करके ये फॉर्म फिल करना है जो चैट बॉक्स में मैंने दी है. उसके बाद यू आर फ्री. आप अपना टाइम जैसे यूज़ करना है वैसे यूज़ करके वो प्रैक्टिकल का जो कंटेंट मैंने आपको दिया है गूगल क्लासरूम में, वो कंटेंट वहाँ से कहीं गायब नहीं होने वाला है. उसको ओपन करके आप राइटअप करना शुरू कर दीजिए. और जब भी आपका कंप्लीट हो जाता है, उसके बाद आप क्या करो, वो प्रैक्टिकल का अटेंडेंस फॉर्म जो दिया है उसके अंदर वो लिंक ऐड करके सबमिट कर दीजिए. दैट्स इट, दैट इज़ द एंड ऑफ ए प्रैक्टिकल. तो एज़ हमने प्रैक्टिकल 11 बजे से पहले ही मैंने डिस्कशन आपसे किया, तो आपका काम ज़रा जल्दी हो जाएगा. और उसके बाद आप जैसे आई थिंक यस्टरडे दिनेश सर हैज़ टोल्ड यू अबाउट his schedule about conducting the class. Okay,